Susan. So I'm very thankful to Section 33 Applied Physical Sciences for pushing through my election to the Academy, and I'm really honored to represent my second home now in the Academy, which is Class 2, the Biological Sciences. So the microscope and the telescope were developed contemporaneously 400 years ago in the early 17th century. And immediately they started to reveal new worlds above our heads, as well as new micro worlds all around us and below our feet. But over the next 400 years, the telescope made steady progress and increasingly expanded our view of the universe until today we see our true insignificance. But the optical microscope, although it got off to a very fast start with guys like Hooke and van Leeuwenhoek, by the end of the 17th century, van Leeuwenhoek was already being able to see bacteria with these microscopes. The bad news is that when he died, he took the secrets of his lens-making ability with him, and it was 200 years before anybody had a microscope as good as his. And so finally, by the late 19th century, Zeiss worked together with a physicist, Abbe, and a chemist, Schott, and together they were able to create finally a microscope that got and slightly surpassed van Leeuwenhoek's limits. But they actually found that there was actually a fundamental limit to how small you could see in the optical microscope, which is about half of the wavelength of light. We call that either the Abbe limit or the diffraction limit. Therefore, from the late 19th century up until the 1980s, there was actually very little additional evolution in the microscope. In contrast mechanisms, so forth, yes. But really, most of the microscopes biologists would use in 1980 were little different from the microscopes that Zeiss was producing in the 1880s. So what changed? Well, in the 1980s, basically several different random technologies assembled together to ignite a revolution. The first of these started with the transistor and the integrated circuit, which led to computers, and in particular by the 80s, computers in every pot, that everybody could have access to them. So you could control your microscopes, you could do image processing, you could do data analysis. In addition, the semiconductor technology spun off into new cameras and so forth that had amazing sensitivity to see single photons, to be extremely fast, and have all those other benefits. The second innovation was the development of the laser, which in the early 60s finally came on the scene, but was really in the hands of many biologists by the 1980s. And the ability to have these high brightness sources of very pure colors and being able to sculpt how you use that light because of the coherence and the phase control that you had of the light was important. And the final thing for biology in particular was the development of fluorescent probes. First, although fluorescence has been around for a century, really antibodies and antibody labeling first, but in particular in '94 when Chalfie first used GFP and C. elegans, and since then fluorescent proteins have been a mainstay of imaging and biological discovery. So these three very disparate fields came together to really ignite a revolution which is continuing to this day. So I had the dumb luck to basically be able to get in on the ground floor of this revolution when I went to grad school in '82 and started working on something called the near-field microscope. So the idea was to try to see if we could get around Abbe's limit to the resolution limit by making a sharp probe that would have a small hole in it, and then we would pass light through that hole to act as a little nano flashlight that we would drive around the sample and get an image. And so I worked on that for 6 years and eventually got what I wanted out of it, which was my thesis, and we poked slightly beyond the diffraction limit, and I got the other thing I wanted out of it, which was a job at Bell Labs. So I then had my own lab in '88, and we further refined the technology over the next 5 or 6 years, and at one time we had the world record for data storage density. We were the first people to do super resolution fluorescence imaging of fixed cells. I was the first guy to see single molecules and their positions to a 50th of a wavelength of light back in '93, and with my buddy Harold, we did the first nanoscale spectroscopy of semiconductor structures at cryogenic temperatures. But by '94, I had been doing this technique for a dozen years, and I was really disillusioned. The first problem was, as every technology has, it has its limitations. And with near field, it's the light that comes out of that little hole spreads really rapidly, so you have to be extremely close to the surface. My dream when I entered grad school and heard about near field was to make an optical microscope that would allow us to look at living cells with the resolution of an electron microscope. But a cell is a heck of a lot rougher than this 25 nanometers where you lose the resolution. And so it was never going to happen with that. The other thing is I had a hard lesson in technology development. I've now worked on probably 6 different technologies over the course of my career, and they all follow this curve. This is just like Dr. Hooper's curve, you know, where it starts like this and it goes like this, and then, like you said, the hype drove me nuts. So once we started to have success with this microscope, many hundreds of people start to jump into the field. Most of them have never been in that field, don't know what they're talking about. The signal of the field stays the same, and the noise goes to infinity, so the SNR goes to 0. And so I've learned over time that every technology is like a newborn baby, and you invest so much of your blood, sweat, and tears in it, you love it like your own child. But just like your own child, you think that it's going to cure cancer or become president, but in the end, you're just happy if it stays out of jail. And near field just barely stayed out of jail because of these limitations. But whereas I thought it would end up here, it ended up here, and everybody else at the same time in the whole academic world is saying it's going to go there. And it just drove me nuts. I really felt like every good paper we published was the justification for 100 pieces of crap to follow in its wake. And everything I had done was a net negative to society. And so I quit. And I didn't know what I was going to do. I was pushing around my daughter in a stroller and realized, inspired by some experiments with near field, that there might be a different way to beat the diffraction limit by taking all of these blobby images of fluorescent molecules that normally overlap, isolating them, for example, based on their colors in a higher dimensional space, and then finding the center of each fuzzy ball to find their positions. But it was going to be too much work, and I was too disillusioned to want to pursue that idea. So instead, I worked for my father's machine tool company, worked on a technology I was very proud of. Over 7 years, I developed it, and in the end, it was a complete disaster when I sold only 2 of them. And so in 2003, I quit working for my dad, and now I was really up against the wall. So one of the things I decided during that midlife crisis was that I really wanted to get back to science because I miss building things with my own hands and following my curiosity. My best friend Harold had gone into industry and felt similarly, so we started casting around for an idea or something to do. We went to Florida State University, where we learned that there was a new type of fluorescent protein that had just come on the scene, which is initially dark until you activate it, and then it becomes fluorescent. And we realized, oh my God, this would make that idea that I had pitched before about isolating the molecules and then finding their centers to work. So if we turn on just a few molecules at a time by dialing down the light that does that, then when you only get a few fuzzy spots, and you can find the centers of those fuzzy spots, you turn on another subset, another, and another. So that's what we call photoactivated localization microscopy of palm. Our problem was we were both unemployed, but that didn't stop us. So when I left Bell, I told him to go to hell, but when Harold left Bell, he was able to take all of his equipment with him. So we took that out of the storage shed and put it together in his living room. But we still didn't know anything about biology, so we teamed up with Jennifer Lippincott-Schwartz and George Patterson, who invented PAGFP in the early 2000s, and we took the microscope to her lab, and with George's help, within a month or so, we've started getting images like this, where we could turn on molecules and lysosomes. If you sum up the fuzzy balls, you get a diffraction limit image, and if instead you find the centers, you start to build something else. That something else, after 20,000 frames, goes from this to that, or to better appreciate the resolution gained from that to that. So with this palm technique, basically you could go 10 times beyond Abbe's limit with a microscope you can build in your living room. And so therefore it immediately took off and became a big deal, and it's what got me to Stockholm. So, but the 2 papers the committee cited for giving me the prize were both done while I was unemployed. And with no funding whatsoever. So, Anyway, so over the next couple of years, Harold and I got jobs at Hughes Medical Institutes, Janelia Research Campus. We lived and breathed Palm, did applications such as showing that there's a stochastic model of self-assembly that can predict the positions of chemotaxis receptors in bacteria, show that one method of silencing gene expression is the spatial segregation of core promoters from the genes themselves. And in Harold's case, he made this exquisitely sensitive 3D microscope that with Claire Waterman at NIH, they were able to deduce the total vertical architecture of different proteins and focal adhesions, the points of contact to the cell, to the extracellular matrix. But probably one of the most important applications of PALM has turned out to be something called SPT PALM, where you photoactivate subsets of molecules and watch their diffusion and their assembly into larger structures. A modern example of that is this work by James Liu at Janelia, where he's looking at Huntington's disease and the Huntington mutant, which creates these aggregates inside of the nucleus. And what he found is that both the wild-type Huntington protein as well as various transcription factors are slowed and sucked up by these aggregates. And so this impedes their search process along the DNA and hence screws up gene expression and then has these downstream consequences for Huntington's. So by 2008, of course, super resolution once again became a huge field with hundreds of people in it, and I felt like I was living the same bad nightmare all over again, okay? Because most of the people going into it don't know what they're doing, and they overhype it like crazy. And as good as the techniques are, and they have applications, they have many limitations again, you know, staying out of jail, okay? So with super resolution, the problem is you need extremely high labeling densities to decorate finely enough the proteins or the structures you care about seeing, and that's a real challenge for the biologists. Another problem is if you're going to look at fixed cells, chemical fixatives are known to completely change the ultrastructure, so you're looking at an artifact even from the very beginning. The other alternative, and really the thing I wanted to do all along, is to do live imaging, but the two methods to share the ward, the STED technique developed by Stefan Hell and the localization by me and WE, they require extremely high intensities, anywhere from 10,000 to 1 billion times more than what light evolved under, and they're extremely slow compared to the speeds at which things move in cells. So they're really not, except for that SPT PALM example, great live tools. And so by 2008, I was again contemplating quitting, but fortunately I was a little more mature now and stayed my ground at Janelia. And I realized, you know, that you have to listen to the customer. That's what I learned from the failure of my dad's company. And the biologists are my customers. And I learned from those people that a physicist thinks of a microscope in terms of resolution, but a biologist knows there's a lot more that you have to keep along with that resolution in terms of speed, no damage to the specimen, ability to image deep. And so from that time in 2008, and that epiphany, the focus of my group is to develop there's no one microscope that can do it all. There's always trade-offs between those things. But that's good news for me as a technology developer, because now I try to develop different microscopes to serve different parts within that tetrahedron to help biologists answer questions they couldn't answer before by previous technologies. One of those technologies is the third method of super resolution, modern super resolution, called structured illumination, where you put on a grading of illumination instead of uniform illumination, which creates these beading patterns, these moire fringes that tell you about the information in the sample. This was invented by Mats Gustafsson while he was at UCSF, and then he came to Janelia, and he was my colleague, but he died of a brain tumor in 2011. And so I've been kind of trying to extend his legacy in this area, in one way with a very fast SIM microscope, in this case to be able to look at the dynamics of the endoplasmic reticulum at up to 50 frames a second and 100 nanometer resolution. We've applied that with John Hammer's group at NIH to look at how myosins interact. So myosin 2 has 3 different isoforms, and these myosins group together to form these bipolar filaments. In this case, we're looking at two different types of myosins, myosin 18 and myosin 2, and they also group together as they were able to discover with the SIM, and that furthermore, the myosin 18 acts as a regulator of the length of the bipolar filament and provides an additional means for ways of these filaments to anchor to other structures inside of the cell. So we also looked at the live formation of these bipolar filaments and found that while here you're seeing additional de novo nucleation of a microtube, or a bipolar filament, and it becomes mature here, but oh, look at that. What happened? It split off the other one. And if we look a little longer, you're going to see another one split off. And so what we found, you can see a whole cascading production from one progenitor here of this stuff, is that myosin filaments largely form. They can form de novo, but it's much easier for them to actually be nucleated from existing myosin filaments, and that's what happens in the cell, all revealed with SIM. So in my opinion, SIM is perhaps, along with localization, but probably even more than localization, the tool that's of most use to most biologists in the super resolution realm. But its primary limitation that it's limited to only twice beyond Abbe's limit. And that was actually the reason why the Nobel Committee stated that it shouldn't be part of the prize. Well, if that's the case, then the focus of my group is to try to prove them wrong and show that we can do SIM beyond that limit. So one way is by using insanely high numerical aperture objectives that you can buy that push the resolution to 82 nanometers. And then in this case, we worked with Tommy Kirchhausen to look at the role of actin in clathrin-mediated endocytosis, or how cargoes are brought past the plasma membrane into the cell. We also developed a method of using the same photo-switching tricks that we used in the Palm approach to use two standing waves of light, which allows us to go, instead of twice beyond the diffraction limit, to 3 times, and hence get us down to 60 nanometer resolution. At that point, in 2014, I really felt I had finally met the goal I had set for myself in 1982 to be able to look at the living cell with resolution close to the electron microscope. The problem with these methods I've just discussed, though, is that they only work close to the plasma membrane of the cell, and you really want to be able to see the entirety of the cell. So I wanted to set for myself a goal to make a microscope that would be as transformative for studying the 3D dynamics of the cell in terms of its speed and its non-invasiveness as Palm was transformative in terms of resolution. So that ultimately led to something we called the Lattice light sheet microscope, which uses non-diffracting beams like you have in a supermarket checkout scanner, or optical lattices that they use to trap and cool atoms near absolute zero, but instead make these ultra-thin light sheets that we could sweep through the cell and plane by plane get our image. And this microscope, we've had over 50 groups come to use this microscope in my lab, and every one of them goes away with big smiles and 10 terabytes of data. But we never hear from them again because they have no idea what to do with 10 terabytes of data. But in any case, it was nice to win the Nobel Prize for Palm, but I'm morally certain that this microscope will be the high watermark of my career, because it just reveals, I've never felt more like Galileo than I have with this microscope. And every week and every new collaborator is an adventure in which we see the cell or the organism as we've never seen it before. So one of the ways that microscope has turned out valuable was it forced me back to super resolution, because it's exquisitely good at being able to get great signal to noise of seeing single molecules even in dense, complicated environments. And so now with various single molecule techniques, we can extend that sort of Palm type localization technologies even to zebrafish organs with sort of, nanoscale resolution. But my real interest is, after 2008, I said, damn it, I've looked at dead stuff too much. I'm going to just focus on live stuff. And the Lattice is a great live tool. Here's an example with Tommy Kirchhausen's group where we were looking at genome-edited cells, and again, clathrin-mediated endocytosis. But now looking across not just the bottom of the cell, but all of the cell. And what we found is that at the very leading edge, where you see this sort of street sweeper leading edge going, this treadmilling edge, is that endocytosis is shut down on the top surface. And so what Tommy believes is happening is now there's an imbalance because exocytosis is still happening to deposit membrane there, and that excess membrane is what's exactly creating these ruffles. And then right beyond that range, you get a ton of exocytosis as it starts sucking back in that extra membrane as it comes back around. Tommy also worked to calibrate the sensitivity of his microscope to single molecule sensitivity and was able to show the relative roles of clathrin-dependent endocytosis versus clathrin-independent by looking at Shiga toxin. And what he found is that these clathrin-independent events are far more common, but because the cargoes are so much bigger in the clathrin-dependent, it still results for about 85% of the amount of cargo that's brought in. And so we can extend not just to single cells, but multicellular. So here we're looking at the interaction of a T cell with a dendritic cell and looking in 3D at the formation of the immunological synapse. Here we're looking at a protein which leaves a mid-body remnant but clearly localizes to other things in the C. elegans embryo. Here we're looking at a whole Drosophila embryo, and halfway through embryogenesis, these epithelial cells come and close up over these earlier cells and create what's known as the purse string structure with Dan Kehart's Duca group. But we have a problem in that as you try to go deeper into multicellular specimens, the light sheet gets scrambled by the different refractive index inhomogeneities, and the light that gets out gets scrambled as well, just like if you have water on your windshield scrambling the light in front of you. So the good news is astronomers had the same problem 50 years ago because the atmosphere does the same thing to the objects they want to see. And so they developed a technique called adaptive optics, where they shine a laser in the stratosphere to create an artificial guide star, which then comes back into their telescope with their dim astronomical object, and they use a computer to change the shape of a mirror to cancel out these aberrations and go from that to that. So as I say in my talk I give on the historical development of astronomy and microscopy, microscopy is the retarded stepchild of astronomy because we always steal everything from astronomers 50 years after they do it. And so we're doing that again with adaptive optics. This is something my wife is also expert in. And so here's a zebrafish embryo here. We use a 2-photon laser to create a little spot of fluorescence to go into our sensor, to go to our deformal mirror. This is looking in the hindbrain region of the fish, and you turn off the adaptive optics, you turn on, and basically this is just diffraction-limited imaging, but we're recovering it. So any biologists in the audience who think they're doing what their microscope says on the barrel in terms of resolution when they do multicellular imaging, I'm here to tell you you're wrong, okay? And you need adaptive optics. So here's some examples of some work we did. This is looking at a fair fraction of a zebrafish brain 72 hours post-fertilization. Looking at a sparse subset of neurons. Here we're looking at mitochondria in a soma that's 150 microns deep. And with the adaptive optics, we can see all of those mitochondria as well as if this were a cell on a cover slip. In this case, we're going deep in the hindbrain, and then we're going to turn off the adaptive optics, and you would see what you would see in a normal microscope. So the very fact that we can now see in the whole organism the cell just as it should be on a cover slip and get back to diffraction limit has led to the new focus of my group, which is that, look, I was trained as a physicist. I'm certainly no biologist, despite the fact that I'm representing biology, and I don't know how that happened. But anyway, so, we learn on our mother's knee about Heisenberg tells us that if you do an experiment, the observer perturbs the system. And it's my opinion that we have never seen the cell on its own terms in its true physiological state for at least 3 reasons. One, fluorescent proteins are great, but until recently, we've looked at them in cells where their expression has been uncontrolled. But now with genome editing techniques such as CRISPR-Cas9, we can look at them at endogenous levels. The second problem is that the microscopes we typically use, like confocal microscopes, throw far too much light at the specimen and actually perturb the physiology if they don't actually kill the cell. And so techniques like lattice light sheet finally give us the ability to look at cells non-invasively for long-term at high resolution. And the final thing is too much of what we know about cell biology comes from looking at cells on cover slips, and they damn well didn't evolve there. And so we need to be able to look at them in the whole organism, where there's all the environmental cues from the surrounding matrix that determines what's going to be expressed. And for that, we need adaptive optics. And so this is some of the initial work now of combining the adaptive optics with the lattice light sheet. You could see the AO get turned on. These are dextrin particles. This is a fluid-filled cavity in the ear of a zebrafish. This is a little neutrophil that's now coming around and trying to sweep up all of the garbage in the space or any other bacteria. This is the blood vessel here and so forth. This is a 3D representation of a similar data set here. So this is all this sort of mesenchymal space and this perilymphatic space. And there's our little neutrophil going around cleaning things up and so forth. So now we really do have the tools to get really great resolution inside of the whole evolving organism. This microscope is hot off the presses. We're going to spend the next 6 months working with biologists to try to take it through its paces, just as we did with the original lattice scope. And so with that, I'd like to conclude and just say that, I'd say the revolution continues. I mean, every day there are new technological developments, and the thing I've learned is that science depends on technology, technology depends on science. It's a yin-yang kind of thing, and every time there's a new little widget on the scene, it's another thing to allow us to go higher up on this exponential curve of this revolution in microscopy. But if there's one thing you should take from this talk, is that this is a very interdisciplinary activity, right? And really, I think that's one of the beauties of the place I work at Janelia Farm, because we put all of the disciplines within the same building. Chemists, physicists, computational scientists, geneticists, etc. And we all work together to solve problems for biology. And that's an incredibly powerful model that I wish the academic community would try to learn from. Thank you.